In this section, we will discuss the humoral arm of adaptive immunity. In this section, we'll cover the branch of adaptive immunity known as humoral immunity. Remember that humoral immunity is all about B cells. So here in this slide, we see how B cells are clonally selected, or the process by which a B cell becomes mature and then activates and proliferates in response to its given antigen. Remember that B cells start out in the bone marrow coming from stem cells. These stem cells will differentiate through the lymphoid lineage into either B cells or T cells. Now, if it's a B cell, then it will start to rearrange certain genes which will then provide it a B cell receptor. This B cell receptor, again, is specific for an antigen that it can find in its native conformation out in the environment. And so once these B cells are fully mature, once they have their B cell receptor in place, they're released into the blood, where they go through the blood and the lymph, visiting various lymphoid organs in search of their given antigen or their specific antigen. So once they encounter their antigen, then they start to get active. So we see through these first couple of steps, we see the stem cell differentiating, then the gene rearrangement, which provides the specific B cell receptor, with each B cell getting a unique B cell receptor. So in this case, the receptor on 1 would be different than 2, which would be different than 3, and so on and so on. Now once these cells are released, here in this case, let's say that the B cell number 2 happens upon its antigen. You're infected with a virus that happens to have a protein specific for this B cell number 2. Once B cell number 2 sees this protein, it will become activated and it will start to respond. Note that only 2 is responding here. 1 doesn't respond, 3 doesn't respond, just that one that is specific for that antigen. It starts to proliferate. That makes sense since it's the only one that's specific for whatever it is that you are infected with. So this B cell number 2 becomes active, it starts to proliferate. As it gets signals from T cells, it will start to differentiate. Now you will see what the end result here is these plasma cells. A couple of things to note about these plasma cells. Notice the upregulated endoplasmic reticulum. Again, remember that these are antibiotic factories. They're not involved in finding antigen, they're not involved in presenting antigen to T cells, they don't care about any of that. Plasma cells want to make antibodies. So upregulated endoplasmic reticulum, and also note that they don't have any B cell receptors on their surface. Again, that time is past. It's not about finding the receptor anymore. It's not about finding the antigen. It's about making antibodies. So, in a textual format, this is kind of what we just saw. Naive B cells recognize their cognate antigen in the lymph nodes. Effector B cells are activated in situ, meaning that they're activated right there on the spot. They clonally expand and they secrete soluble antibody of the same antigen specificity. So the specificity that's on the B cell receptor is the exact same specificity of the antibodies that these plasma cells start to pump out. With the help from T helper cytokines, these B cells can perform what we call antibody isotype switching, and we'll get to that in a little while. Plasma and memory cells are formed at the end of the day, with plasma cells being the antibody factories, memory cells being there for secondary responses. Some stay in the lymph nodes, others migrate back to the bone marrow. The source of antigen is eliminated in the effector cells. Those plasma cells again go through a contractual phase where they will die off. So B lymphocytes are formed in the bone marrow. They will continue to mature in the marrow and acquire specific B cell receptors and commit to single antigen specificity. Like we saw in the past couple of slides, every B cell has a very specific B cell receptor, and it will only recognize a specific antigen. B cells that do not recognize self-peptides are permitted to survive, so we have a level of control there to prevent against autoimmunity. B cells that express high affinity receptors for self-peptides are given a chance to try again. They undergo a process called receptor editing, where they can manipulate their B cell receptor in hopes of coming up with a B cell receptor that would be specific for a foreign antigen and not a self-antigen. If this receptor editing is unsuccessful and the B cells are still specific to self-peptides, then they will undergo death as part of our central tolerance mechanisms. The naive B cells that make it through this process, that survive and are not self-reactive, will then migrate to the lymph nodes, where they will set up shop and wait for the lymph to be filtered through and try to detect antigen. So here we have a very basic structure of a B cell receptor, or an antibody, also known as an immunoglobulin. And here we see very specific vocabulary terms here, paratope and epitope. We've discussed epitope in prior sections. Remember, the epitope is the specific portion of the antigen that the antibody binds to. So the epitope is part of the antigen. The paratope is the part of the B cell, or the part of the antibody or the B cell receptor that will bind to the epitope. So the paratope on the B cell receptor binds to the epitope on the antigen. Some contrast between T cells and B cell antigens. Remember that T cells can only see proteins. All that they're looking for are these peptide chains that have been processed and presented to them in the context of an MHC molecule. B cells, on the other hand, see proteins, but also see polysaccharides, they can see lipids, they can see small molecules such as DNA. Antibodies have a much broader range that they can bind to. There's no need for antigen processing. They can detect antigen in its native conformation. And they tend to look more for external epitopes, epitopes that are exposed to the outside, hydrophilic type of epitopes, versus T cells that they can really get down since this antigen is processed and find internal epitopes, epitopes on the inside of antigens. So here's the question. A researcher is studying specific immune responses to an antigen by incubating it with several different cell types. He notices that the antigen will cause activation of B cells alone, but not T cells alone. However, when the T cells are incubated with antigen-presenting cells in the presence of the antigen, they become activated. What type of antigen is most likely present? A whole protein in native configuration, B, a whole protein that has been denatured, C, a whole protein cut into 13-mer peptides, D, a whole protein cut into nonomer peptides, or E, a whole protein cut into tetramer peptides. The answer is A, a whole protein in native configuration. Why is this? Because in this specific example, we see that when the antigen is incubated with B cells, then they will be activated, but not T cells. Remember that B cells like that native antigen. They look for the native antigen. They don't want anything processed. And even some B cells look at the tertiary structures of proteins. So things that have been denatured can also affect whether or not a B cell can bind. So here what we're looking at is a whole unprocessed antigen. Those are what the B cells look for. Hence, the correct answer is A. So what's the mechanism of humoral immunity? What do these antibodies do? Well, they attach to the surface of extracellular microbes. In and of themselves, they're not cytotoxic. They can't kill bacteria. They don't have any properties like that. These are the things that they can do, however. They can act as opsonins to enhance phagocytosis. They will bind to a prokaryote, and then macrophages and neutrophils can come in that actually have receptors for these antibodies. They will recognize the antibodies, and then in that aspect, the antibody is telling the phagocyte what it needs to destroy. Antibodies can also activate the complement pathway to lyse cells, specifically the classical complement pathway. Antibodies can agglutinate bacteria, which also enhances phagocytosis. They can bind to microbial surface receptors and prevent their adherence. If this is the case, then they're known as a neutralizing antibody, in so much that they can bind to certain proteins on viruses, for example, that are involved in adherence mechanisms and prevent them from being able to infect cells. They can also bind to and neutralize microbial toxins and serve as an antitoxin in that aspect. So here we have a little more detailed structure of the B cell receptor or the antibody. Notice that these have many different loops in them, which are called immunoglobulin folds, that are held together by these disulfide bridges. Note that there's 4 separate polypeptide chains, 2 heavy chains that run down the middle, and 2 light chains that are on the outside. Within each of these heavy and light chains, there's variable loops and there's constant loops, denoted by V or C. So the heavy chain has 1 variable loop, and in this case, 3 constant loops, and the light chain has 1 variable loop and 1 constant loop. It's also important to note that within an antibody, the heavy chains, both heavy chains are exactly the same, and both light chains are exactly the same. They were encoded from a genetic standpoint, and every heavy chain after the B cell receptor structure has been determined will be exactly the same. Every light chain will also be exactly the same. That's why we can say that every antibody that B cell produces is going to have the exact same specificity. Note that the heavy chain constant domains make up what we would call the bottom part of the Y of the antibody. This is what's referred to as the Fc portion of the antibody, And when we talk about Fc receptors on cells, this is the portion of the antibody that we're talking about. Another important point here is to note that this hinge region in the middle of the antibody promotes flexibility and better binding. It allows the antibody to kind of tilt one way or another to allow it to bind to more than one thing. This also comes into play when we talk about papain and pepsin. So here we have the genes, getting down to the basic genetic level of how we determine what the specificity of a B cell receptor or an antibody is going to be. We have to have a way for our B cells to be able to recognize the millions of different antigen combinations that may be out there. So one mechanism could be that we would have millions of different genes within our B cells. That's not really feasible. The mechanism that our body has come up with is quite ingenious, and we will talk about that now. At the germline level, we have different regions of genes that we have to concern ourselves with as far as the heavy chain synthesis goes. These regions are the V region, the D region, and the J region, which you can see here on the top of the slide. Now, there are several different genes in each of these regions. There are many V's, many D's, and many J's. And what the B cell does while it's in the bone marrow undergoing maturation processes is it rearranges and it grabs random V's, random D's, and random J's and pushes them together in order to give some kind of random combination. But this random combination is where the specificity of that B cell comes from. And each B cell is going to develop independently of the B cell next to it. So one B cell is going to grab certain VDJ, another B cell is going to grab a different VD and J, and that's how you come up with the different specificities among the B cells. So let's kind of walk through this step by step. We're starting with the D and the J regions. We can see there at the top of the slide where the 1 is. In this process, to get this process started on the heavy chain, the B cell is going to grab one of the D genes from the D regions, let's say DH7, and one of the J genes, let's say JH3. And it's going to move those genes right next to each other, the DH7 and the JH3. Everything in the middle, everything in between those, is actually going to be excised out of the DNA so that we now have that DH7 right next to the JH3. The next step is that the B cell will grab one of these V genes from the V region and it will hook it up to the other side of that DH7. So in this case it takes the VH1 and it hooks it up to the other side of that DH7, again looping out everything that's in the middle. So now we have these 3 genes that have been placed side by side, VH1, DH7, JH3. That's going to make up our variable loop on the heavy chain. That's part of what gives us the specificity. That's going to be the specificity part of the heavy chain, that VDJ recombination. And again, this is at the DNA level. So as we go further, as we move from a B cell receptor to secreted antibodies, that change has been made at the DNA. So all of the heavy chains that are coming out of that B cell are going to have this recombination, this VH1, DH7, JH3. So as we move to the next step, we have our V loop. Now we have to get our C loops. That comes from these constant genes, these C genes that are tailing after the J region. We see C mu, C delta, C gamma, C epsilon, and C alpha. These encode for different C regions and will give us different properties of the antibodies, which we'll discuss later. We know that we have IgM, IgD, IgG. Those come from these different C genes. We'll discuss that a little bit later. The step as far as the genetics are concerned is that it takes this VDJ recombination and it's going to hook it to the first constant gene that it comes in contact with. In this case, it's the C mu. In the heavy chain, in a naive B cell, it's always going to be that C mu. It's going to be the first one. And that's why all antibodies, or all B cells, the first antibody that they make is an IgM. So it takes this VDJ region, it hooks it up to this C mu, and it loops out everything that's in between. So now at the mRNA level, we have our VDJ recombination. That's going to make our V loop. We have this C mu gene. That's going to encode all of our C loops. That makes up our constant region. We give it a 5' cap and a poly A tail. Then we translate it, and now you have a mu heavy chain, a heavy chain that's going to encode for an IgM type of antibody. In the next step, we have to now form our light chain. Once we have the heavy chain down, we have to form our light chain. Now, light chains can come in 2 flavors. They're either kappa or lambda. They're just different genes. They both do the same thing, different gene regions within your genome. Now, as order would have it, your body is always going to try to rearrange the kappa chains first. So once it gets a heavy chain that's productive, that's functional, then it's going to try to rearrange one of the kappa chains. Remember, you have two alleles, just like every other gene. You have one from your mom, one from your dad. It's going to pick one of those kappas and try to rearrange it. Now, if it can rearrange it, then you're done. I've got a heavy chain, I've got a light chain, and I'm good to go. If I can't rearrange the first kappa, then I move to the second kappa. And if I can rearrange the second kappa successfully, then I'm done. If I can't, that's when I move into lambda, and I'll try lambda. But that's the sequence. So heavy chain first. If you have successful heavy chain, you go to kappa. If you can do kappa light chains, then you're done. If for some reason you can't successfully recombine kappa chains, then you go to lambda chains. That's kind of your last resort. So let's look at these kappa chains. Very similar setup to the heavy chain with one exception. If you'll notice, there's no D region here. Other than that, it's exactly the same. You're going to grab one of the V genes and one of the J genes, and you're going to stick them together. In this case, it's V kappa 2, J kappa 3. You're going to stick them together, and everything that's in the middle is going to be looped out. And that's going to be your V loop for your light chain, that V kappa 2, J kappa 3. That's the specificity on the light chain. So now we've got our paratope. We've got the heavy chain V loop and the light chain V loop. That's what makes your specificity. All we have left to do now, we have to loop that VJ recombination over to the C kappa gene, which is the constant loop. We'll loop out everything in the middle, just like we did in the heavy chain. Now we've got V kappa 2, J kappa 3, right next to the C kappa. There we go. We put a 5' cap, a poly A tail, we translate it. Now we have our kappa chain. So if that's successful, we've got our heavy chain now. We're going to make 2 of those. We've got our light chain now. We're going to make 2 of those. And now we have an antibody. 2 heavy chains, 2 light chains. If the kappa chain fails, then we move over to the lambda chain. And it's worth noting here that the lambda setup is a little bit different than what we're used to. Instead of having a V region, a J region, and a C region, each J gene within the lambda region has its own C gene accompanying it. So that once you picked the J for the recombination sequence, you automatically get the C that goes with it. So here we have the V lambda 1 hooking up to the J lambda 4, and then the C lambda 4 automatically goes with it. Other than that, everything's the same. Everything gets looped out. Now you've got your recombination, you convert it to mRNA, and you make your lambda chain. So, successful lambda, again, now you have your antibody. You have your heavy chains, you have your light chains, you can make an antibody. If neither lambda is successful, then what happens is the B cell just undergoes apoptosis. Here we have another question. A researcher is studying the structure and function of B cell receptors. She notes that the intermembrane portion of the receptor is structurally similar to the Fc portion of an IgM antibody. The decision to make this specific antibody type is made during which stage of recombination? A. DJ joining on the heavy chain. B. VDJ joining on the heavy chain. C. VDJ-C joining on the heavy chain. D. VJ joining on the light chain. Or E. VJ-C joining on the light chain. The answer here is C. VDJ-C joining on the heavy chain. So we know from this question it's asking about the Fc portion. It says that we've got something that's similar to the Fc portion, and what stage is this decision being made? Well, we know that the Fc portion, if we think back to our diagram, all comes from the heavy chain. Light chain has nothing to do with Fc region, so we can get rid of D and E right there. Now, between A, B, and C, where is this decision made? Well, it comes from the C gene. Remember, the VDJ just makes up that V loop, that first loop that we see on the heavy chain. The rest of it, which actually would make up the Fc portion of the antibody, comes from the C gene. So the decision is made during the VDJ-C joining on the heavy chain. In these next 2 slides, we're going to talk about papain and pepsin. From a medical standpoint, papain and pepsin you really need to know for the boards, and then you can forget about it. But because we're studying for the boards, we've got to know this. So we'll talk about these 2 different chemicals, what they do to antibodies specifically, and then how they can be interpreted and how they can be used by the boards in the question. So let's first look at papain. Papain is a proteolytic enzyme. that can cleave an antibody right at the hinge region. So we can see here on the slide what it does is it breaks this antibody up into essentially three different parts. Two what we call Fab fragments and one FC fragment. The Fab fragments are antigen binding portions of the antibody. Notice that they're the fragments that have that antigen binding region, or the paratope, that VH and VL. So they can both bind to antigen. And then the FC fragment is that constant part of the heavy chain broken out. Remember that these things can bind to FC receptors on macrophages or mast cells, for example. So this is what happens with papain. You get two Fab fragments and one FC fragment. Contrast that with pepsin. Pepsin is a proteolytic enzyme that leaves the hinge region intact but essentially destroys the FC portion. So at the end of the day, what we have here is what we call a Fab prime 2 divalent fragment. So notice that both of the Fab fragments are still stuck together at the hinge region, and we call that a Fab prime 2 divalent fragment. The FC portion has been destroyed. So we have one Fab prime 2 fragment and nothing else with pepsin. And with papain, remember we have two Fab fragments and one FC fragment. So how does it get used in the boards? Here's an example. Your biotech company has developed a new line of compounds prepared by proteolytic digestion of purified anti-nickel human IgG antibodies. Compound A is prepared with papain, and compound B is prepared with pepsin. Which one of the two compounds will be most useful as a positive control reagent for your nickel agglutination assay? A, compound A will be most useful because it contains the FC fragment. B, compound B will be most useful because it contains the FC fragment. C, compound B will be most useful because it contains the Fab prime 2 fragment. D, compound B will be most useful because it contains the Fab fragment. E, compound A will be most useful because it can activate complement needed for the agglutination assay. Or F, compound B because it can still bind to the FC receptors necessary for agglutination. The answer here is C. Compound B will be most useful because it contains Fab prime 2 fragment. So let's go back and talk about how we come to this conclusion. First of all, we have to decide what the function of the assay is. This is a nickel agglutination assay, so we want to be able to stick things together. We want to be able to agglutinate. We know that compound A is prepared with papain and compound B is prepared with pepsin. So with compound A, we get these Fab fragments. These Fab fragments will be specific to nickel. They can bind to nickel, but all they can really do is bind. They can't stick anything together because they're all individual. Contrast that with a Fab prime 2 fragment. Now a Fab prime 2 fragment, which would be the result of pepsin digestion, has two different binding sites on it. So it can actually bind to two different nickel molecules. Now once it's bound to this nickel, you can get another Fab prime 2 that would bind to the same antigen and then an additional antigen. And then you can get another Fab prime 2 fragment that would bind to the same antigen and then an additional antigen, and so on and so on. You can see how you can form a network out of this, and you can agglutinate things. So take home message here. With pepsin, you can agglutinate. With papain, you cannot. So for this answer, you have to have something that agglutinates, and therefore it's compound B, because it contains that Fab prime 2 fragment. Let's discuss antigenic determinants on immunoglobulins. These are some more vocabulary terms that you should be familiar with for the boards. Isotype, allotype, and idiotype. The isotype is determined by the constant region of the heavy chain. This is the type of antibody that it is. So if you hear about IgA, IgE, IgM, IgG, these are isotypes. We remember that those are determined by the FC portion of the antibody. Now allotypes are different. Allotypes are the same isotypes but from different individuals. Because of the way our bodies work, and we're all individual and all unique, the IgG that I make is going to be a little bit different than the IgG that you make. Now structurally, functionally, it's going to be the same, but the little differences that make mine different than yours would be called allotypic differences, in so much that if I put my IgG into you, you might see it as foreign because of those small differences. Those are allotypic differences. Idiotype refers to the portion of the antibody that determines its antigen specificity. Refers to the paratope region. Okay? Every different antibody has a different idiotype. So again, isotype, the character, the FC portion, IgM, IgA, IgG. Allotype, Differences among the same isotype in between individuals, my IgG versus your IgG. Idiotype refers to that paratope, refers to the portion of the antibody that is antigen specific. Here are the different isotypes that you might see. There are 5 different types of antibodies: IgM, IgG, IgE, IgD, and IgA. Note the difference in structure specifically for IgM and IgA. IgM exists in the serum as a pentamer. It's 5 IgM monomers all bound to a protein called J chain, which gives it this nice pentameric snowflake appearance. IgA also utilizes the J chain, but instead of making a pentamer, it only makes a dimer. So IgA exists in dimer as it is secreted out into mucosal secretions. IgG, IgE, and IgD all exist in the serum as monomers, and therefore they do not utilize the J chain. So let's talk about different properties of these antibody isotypes, starting with IgM. IgM, 5 to 10% of the serum type of antibody that you'll see if you do a serum analysis. IgM, some of its claims to fame, is the monomer for IgM is actually a membrane-bound BCR. So the B cell receptor on the surface of a B cell is an IgM monomer. The secreted form, as we just saw, is a pentamer held together by this J chain. IgM is the first immunoglobulin formed in a primary immune response. It's always the first one. Again, going back to our genetics diagram, our genetics slide, remember that C mu was the very first gene. That's the gene that encodes for IgM isotypes. So it's always the first one. IgM is always the first one formed in a primary immune response. Low affinity, again, because it's the primary one, it hasn't had a chance to undergo different enhancements to make it a higher affinity antibody. IgM is also very efficient at complement activation, again, because it's a pentamer. For the classical pathway, it has several Fc regions there for C1 to bind to. It's also good at agglutinating by the same token, since there's 5 different monomers. That's 10 different binding sites that it can bind to antigen with. IgG is typical of a class-switching high-affinity Ig. This is more typical of a secondary response after it receives the cytokine messages from T cells that tell it to become enhanced, that help to tweak the antibody to make it a little more specific, which we'll talk about. This is all what you would think of with an IgG, a high-affinity secondary type of antibody. Very high opsonine capability. It's good at signaling those macrophages. It has the longest serum half-life, and as a consequence, it's the one that's found most abundantly in the blood. You'll see that 80% of the serum antibodies are IgGs. Another unique property of IgG is that it has the ability to cross the placenta. It's the only one that can do this. IgE we hardly ever find in the serum, and that's because of the properties of the Fce receptors found on the surfaces of mast cells and basophils. Remember, when we talked about mast cells and basophils, we said that they have these receptors on their surface to bind to IgE antibodies. It binds to the Fc portion of the IgE antibodies, and it binds so tightly that it really doesn't let them go. Once it has it, it holds on to it. Therefore, IgE is virtually never found in the serum. It's always attached to mast cells or basophils. It's involved in mediating antiparasitic and also allergic responses, which we'll see when we talk about hypersensitivity. IgA is the dominant immunoglobulin that's found in secretions. We see secreted IgA found in breast milk, saliva, tears, mucus. It's a dimerin secretion, and it's a monomer in the serum. So if you're checking blood for IgA, it's going to exist as a monomer. It only attaches to the J chain and becomes a dimer as it's being secreted. In fact, the J chain is involved in mediating the secretion from the vessels out into the mucosal surfaces. IgD is found primarily expressed on B cell surface in conjunction with those IgM monomers, and it's involved primarily in the activation of naive B cells. So, if you would like to selectively measure the levels of serum IgA, which antibody would you use? An anti-isotype, an anti-allotype, an anti-idiotype, an anti-J chain, or an anti-kappa antibody? The answer is A, an anti-isotype. Remember, isotype refers to that Fc region, and since we're specifically looking for IgA, we're looking for a specific type of antibody. We're looking for that isotype, the IgA isotype. So you would use an anti-isotype antibody. Why wouldn't you use an anti-J chain antibody? Two reasons. One, remember that IgM also has the J chain, so you would end up measuring levels of IgM as well. And two, we're looking at serum IgA, and remember that serum IgA does not have the J chain association. Here's another question. A 1-year-old boy is brought to the physician for repeated bacterial infections over the past year and a half. Serum electrophoresis reveals a low level of circulating immunoglobulins. Deficiency of what cytokines would result in impaired antibody production? A. Interferon alpha, B. IL-1, C. IL-4, D. IL-7, or E. TNF alpha? The answer is C. IL-4. In this question, we have a low level of circulating immunoglobulin. So deficiency in which of the following cytokines would result in impaired antibody production? So from the cytokines listed, the one that's most intimately involved with antibody production is IL-4. Another question. Which of the following antibody types are most likely to be found in a newborn infant? A. IgM and IgG, B. IgG and IgA, C. IgA and IgE, D. IgE and IgM, or E. IgA and IgM? The correct answer here is B. IgG and IgA. Again, we can go through these one at a time. We have 4 different antibodies we need to concern ourselves with: M, G, A, and E. So let's talk about them. Would you expect to find IgM in a newborn infant? No, not unless that infant was infected, actively infected, either right before birth or during the birthing process. So you wouldn't expect that to be there. How about IgG? Yes, remember IgG crosses the placenta. How about IgA? Yes, remember that one's found in breast milk, secreted. So G and A should both be expected to be there. And E? Well, E is a secondary antibody that's usually found related to basophils and mast cells, not likely to be found in a newborn infant. So there you have your answer, G and A. So let's look at a primary humoral response in terms of antibody levels, antigen levels, and how this whole thing happens. So here we have a diagram. On the y-axis we're looking at either antibody or antigen titers. On the x-axis we're looking at time. So let's start at the very beginning, time point 0, where we have the antigen challenge. We get infected, virus, bacteria, something like that. During the lag phase, this first phase of the primary humoral response, this is that initial 4 to 5 days that we've talked about where the B cell has to find the antigen and it has to internalize the antigen and it has to present it to a T cell and it has to become activated in and of itself. That takes some time. So during this time, the antigen levels are high, the antibody levels are essentially non-existent because it's taking time to process and get activated and decide that it needs to respond. So after that first initial lag phase, we start the log phase. So this, again, 5 to 7 days after that antigenic challenge, we see the antibody levels start to rise. And notice that those antibody levels are IgM, always IgM first. So we get these antibody levels start to rise, these IgM levels start to come up, and the antigen levels start to drop as the IgM activates complement, as it agglutinates the antigen, as it does the things that it's going to do. We see the antigen levels start to drop off precipitously, and the IgM levels start to rise. So we get to this next phase, the plateau phase, where we have our IgM at our highest levels. Again, this is our primary antibody. This is our low affinity antibody. So out in the serum, out in the blood, out in the tissues, we have lots of IgM. Now down in the B cells, back down to the lymph nodes and in the B cell levels, we have another thing going on. The cytokines that are coming from the T cells are causing class switching, causing those B cells to change, to stop expressing IgM and start expressing IgG or IgA or IgE, one of our secondary antibodies. At the same time, it's also causing these antibodies to become more specific. It's enhancing their ability so that we start to see, as the IgM levels start to fall off, the plasma cells that are making IgM start to die. We get a new set of plasma cells being created that are making IgG and IgA and IgE, those secondary antibodies, those high affinity antibodies. And they start to produce, and we see in that decline phase, a rise in the secondary levels. And notice that those secondary levels actually reach a plateau or a peak that are higher than those IgM levels. So by the end of the response, the antigen is long gone, the IgM is starting to phase out, starting to die off, and these high affinity secondary antibodies start to appear. And those antibodies, like we said with IgE, they last a little bit longer. They allow these cells to be able to contract and at the same time still provide protection in case there's any of that antigen that is still hanging around. Now, at the end of this primary humoral response, those plasma cells, the IgG plasma cells, will also start to die off, and we can see the IgG levels start to drop off a little bit. And it's important to remember at this time, we also have the presence of memory cells, and those B cells, those memory B cells, will live for a long time. That's our memory. That's where we're going to be able to fight these pathogens off again if we ever see them again. So here we have a secondary response, this memory response. We've been talking up this memory so much. Why is memory so important to the adaptive immune response? This is the reason why. So notice the differences here. With antigen exposure at time 0, within 3 days, 2 or 3 days, we get this exponential increase of that high affinity antibodies. Because we have memory B cells already made, the second time we see that antigen, boom, we're off and running. We've seen it before, we know it, we're ready for it, and we don't even have to mess with the IgMs. We can go right to IgG or IgA or IgE, get the high affinity antibodies out into the serum, and we can hit that infection so fast that we don't even become symptomatic. Notice that there's no antigen levels at all anywhere in this slide like we saw in the primaries because the response is that fast. That's why memory is so important. So we get this high, explosive, exponential increase of high affinity antibodies that can take care of the infection before we even know that we had it. Note that about 4 days following that initial exposure, we get another IgM peak coming up. Why would we use this IgM peak? Well, this is actually an IgM response, a primary response to a different epitope on that antigen. So now not only do we have the good response to the first antigen, but we get another primary response to a second or an additional epitope or antigen. So that at the end of this response, we'll actually have now memory cells to 2 different epitopes. So that if we ever get exposed a third time, now we're going to, boom, get 2 responses that are fast. And we'll make a third slow, and so on, so that with each repeated exposure, we get more and more memory cells to more and more of the different epitopes that are present in that antigen, so that the antigens that we're exposed to most in life are going to be the antigens that we respond to best. That's the wonder, that's the brilliance behind this adaptive immunity and this memory function that they have. So here's a question. In a vaccine study, a man is given a vaccination consisting of a specific synthetically derived viral epitope. 3 months later, the man is challenged with the whole virus and the immune response is measured. Which of the following is most likely seen? A, IgG response only, B, IgM response only, C, IgG and IgM, D, IgG and IgD, or E, IgM and IgD? And the answer is C, IgG and IgM. Since we have a vaccination here, this is the whole concept behind vaccines. We have a vaccination, we get a synthetically derived viral epitope, and we expose this person to it so that he will what? Make memory cells to it. We get an initial response and memory cells. 3 months later, he gets the whole virus. Now he's going to have that memory response to the initial antigen. That gives him the IgG burst. And then he's going to make that IgM to another epitope. Because this is a whole virus, there's a different epitope there as well as the original. So we're going to see both that initial IgG burst, and then we'll also see a second primary response come up to a different epitope. Another question, a 26-year-old woman develops a second E. coli urinary tract infection 6 weeks after the first one. The second one is less painful and quickly clears. What antibody is responsible for the fast resolution of symptoms? Is it A, IgA, B, IgD, C, IgG, D, IgE, or E, IgM? The correct response here is A, this is IgA. Here we have a urinary tract infection. Second one is less painful. We have a memory response. Which antibody is responsible? This is a mucosal infection. So the likely secondary antibody that's going to be involved is IgA. Don't get fooled by the secondary response there and automatically associate it with an IgG. Look at what's going on. This is a mucosal type of infection. The best antibody is going to be an IgA as far as resolution of symptoms. So I've mentioned affinity maturation as the process by which these B cells or these antibodies become enhanced or go from a state of low affinity to a state of high affinity. This occurs in the germinal centers of the lymphoid follicles, and B cells are the only ones that do this. T cells won't do this. Also known as somatic hypermutation, this is the phenomenon whereby activated dividing B cells will selectively induce point mutations in the regions of the BCR coding sequence. So it will go along that VDJ recombination sequence that it's made, and it will actually induce point mutations. It will stick nucleotides in there in the hopes of making it higher affinity. And the B cells that bind to antigen the best are then selected to live. And here's an example, kind of looking at it from a scientific point of view. As you can see, starting at the top and working your way from day 7 primary to day 14 primary to the secondary to the tertiary, with each response there's more and more mutations induced in these CDR1, CDR2, and CDR3 regions, which refer to the variable regions on the heavies and light chains. So what we see again is that as the body responds more and more, it starts to tweak its antibodies more and more to get better responses. Here's an example of a T-independent activation. Remember, we said that there's a certain subset of B cells that do not require T cell activation. They live in the splenic marginal zone. So if you go way back, remember that the marginal zone in the spleen is that area in between the white pulp and the red pulp. And these B cells are kind of unique. They're B cells that are specific for repeating monomers, such as LPS. And what we see here is that as the repeating monomers will bind to the binding sites of the surface receptors on these B cells, it will actually cause these surface receptors to move close together. And as they move close together, they will transactivate each other. It's a phenomenon known as transactivation that happens when you bring them close together. Once this occurs, that will signal the B cell that it needs to respond. And as such, it will differentiate into a plasma cell and it will start to make IgM, just like every other B cell. Now, because this is T-independent activation, you don't get cytokine help. Without that cytokine help, these B cells cannot undergo class switching and they cannot undergo affinity maturation. So at the end of the day, you have these IgM-producing plasma cells. You won't get any IgG, you won't get any IgA, you won't get any new affinity or higher affinity, and you won't get any memory cells at the end of the day. All of those things required T cells in order to help. But in the case of making IgM to get rid of this bacteria that has the LPS, for example, it's fairly effective. So another question here. A patient is found to have normal humoral immunity but has minimal cell-mediated immunity. More specifically, the defect is localized to the T cells. Which pathogen is this patient most susceptible to? A. Influenza, B. Conococcus, C. Streptococcus pneumonia, D. E. coli, or E. Clostridium difficile? The answer is A. Influenza. Remember that with minimal cell-mediated immunity, especially minimal T cell-mediated immunity, these are more susceptible to viral infections, intracellular infections. Remember that B cells are involved in extracellular pathogens. And if you look at your choices, B, C, D, and E are all what? Bacteria, extracellular, versus A, which is a virus. That's going to be the one that will be defective response if you lack your T cells. So some humoral response pearls. Remember that naive IgM-positive B cells, meaning those B cells that have an IgM monomer on their surface, will circulate until they encounter antigen. Once they encounter that antigen, they can migrate to the regional lymph nodes, they internalize that antigen, and they can present it on MHC class II. Remember that B cells are also APCs. Once this B cell T helper cell conjugate is formed, the B cell is activated. The B cell can then undergo clonal expansion, affinity maturation, and class switching. So we get higher affinity antibodies, different isotypes, IgG, IgA, IgE. They then differentiate into plasma cells and memory cells. Plasma cells secrete IgM initially in the primary response. They secrete other antibodies during secondary response. And then that second exposure, the anamnestic or the memory response, remember, it's more rapid, higher affinity, higher numbers. This concludes our discussion on humoral immunity. In the next section, we will discuss cell-mediated immunity.